Hi, this is Miss Litton, and this is an honors bio review for the semester final for the spring. Say hi. Hi. All right, so I will go over this review with you. I just want to remind you that all of our calendars for the entire year are at the website. If you just go to the first page and you scroll down, you can see Unit 5 right here in that calendar, and Unit 6, you can see everything that's there, and you can use that to kind of help you prepare for your test. Also, remember there's probably a review. I'm pretty confident there's a review on every single individual chapter. So if there's like one chapter in particular that you're like, freaking out about, then go back and watch that whole chapter review. You don't need to worry about the additional objectives. That is not going to be on there. Okay, those are like too concrete of a fact that you would have to memorize. I don't want to do that. But in the open-ended part at the end, I could ask you a bioethical question that if you've been doing the additional objectives, you might want to pick one or two you could reflect upon potentially, maybe one. That was of interest to you. And then also on the open-ended part of the final would be a question about what we're doing with biomimicry. Okay, so that would be another thing you want to just keep in mind. So we'll go over some big ticket items in each of these that should help prepare you for the final. But remember, wherever you feel like you have weak links, I would keep track of that. You've got a lot of things you're going to be doing between now and the end of school year. You've got things you're still learning in all of your different classes, and you're preparing for your finals. You want to just be really strategic. So as we go through it, you might say, okay, you've been doing collaborative documents this entire school year, which is awesome. But now your final is all about you. So at this point, you might want to open up your own personal private document, or if you're in a group, have a table or something with topics that you especially do need to go over. So when we go over, you could put, Unit 5, Chapter 5, and then if anything sounds scary in that chapter, you write a little note to yourself so you spend a little bit more time reviewing that. Does that make sense? You got a strategy? Okay, so let's start off with Chapter 5. And one of the first things we talked about in Chapter 5, I need my high heels. Okay, was can you diagram the cell cycle and name the parts and what occurs? Do you remember what the cell cycle looks like? I think it's a circle. That is a hideous circle. Do you remember what the bulk of the cell cycle is? Interphase. Good. So I'm going to carve this little mess out right here, which I'm super proud about. And then we can say this right here is all interphase. Do you remember what this is broken down into? You sound very smart to me right now. All right. And then this whole stage right here we would call what? Mitosis probably. And then what are the stages there? Prophase, metaphase, anaphase, telophase. And then cytokinesis. Okay. We'll go with a creative pen. Cytokinesis, maybe no. Cytokinesis has an overlap on top of telophase. Do you need me? I'm recording right now. Okay. All right, so let's talk about what are the key things that occur during G1. Do you remember? Growth. That's good. G for growth. Okay, what else? Organelles, right? Do you remember how we talked about, think about you just finished, if you've done mitosis, one cell became two cells, right? And so you have half as many mitochondria, or if you're a plant cell, half as many chloroplasts or endoplasm reticulum. So to me it makes sense that you would make more organelles right there during G1, okay? S, what does S stand for? Synthesis. What are we synthesizing specifically? DNA. DNA replication. This is when the DNA separates and you make double strands, right? So that whole bit takes place then. G2, what happens? Protein synthesis. Proteins we're going to need for our next steps. Okay, then we go into mitosis. Let's go through the stages. Let me look at the next thing because I forget what I have here. Okay, then we'll apply it there. So then we go through the stages of prophase. Let's go through it visually with a little song, yes? What's the first stage called? What's the whole thing called? Mitosis. What's the first stage? Prophase. The chromosomes become visible. Did I not teach you this? And if you're on my YouTube, it's like mitosis song or something. Okay, here we go. The chromosomes become visible. The nuclear envelope, not the cell membrane, but the nuclear envelope and the nucleolus disappear, radiating microtubules, asters. They meet in the middle at metaphase. Who is they? The chromosomes, right? They consist of two sister chromosomes. Say that again. They meet in the middle at metaphase, away at end. Who's going away? Two nuclei at telophase. So we're rebuilding the nuclear envelope, right? And the nucleolus becomes there again. Two nuclei at telophase, cytokinesis. Okay? That's how one cell becomes two. These are daughter cells. They are clones. They are exactly the same as the parent cell. All right? Now, just that much. Do a self-check. Check. How do you feel about that? You need to review it a little bit more. Tell your bio buddy, how do you feel on that? Clarifying questions you want to tell me? Ask me. I want to tell you right now. I have a question. Yes?
So the centromeres consist of the centrioles and the microtubules, and the centromeres are the whole thing?
Okay, so when you talk about the centrioles, only animal cells have centrioles, primarily, and when you talk about the microtubule organizing center, that's where those centrioles are hanging out, and the microtubule, if you have them, and then the microtubules extend across to help pull the chromosomes apart, or the sister chromatids apart. Okay. More questions? Okay, let's apply it. Let's apply it right here. It says, could you diagram a cell and the stages of mitosis with a cell that has 3 chromosomes, 4 chromosomes, or 5 chromosomes? So let's try that. Let's do 4 chromosomes right now. So how would the cell look like in interphase? What? Just like a solid circle, right? That would be the nucleus, and inside of that would be the nucleolus, yes? But as you move into prophase, what's going to happen? Nuclear envelope and the nucleolus are going to disappear. So then you're going to still see your cell membrane, but your nuclear envelope's going to start to disappear. There's a nucleolus in, oh, I became red. There's a nucleolus in there that's going to start to go away, right? And you're going to start to build up those asters if you have them. The chromosomes are going to start to become visible. So you're going to, at the beginning of it, it's going to start to become visible. Now I'm going to focus just on the nucleus. If we have 4 chromosomes, all 4 chromosomes are going to line up right here in the middle. Why do all 4 chromosomes line up there in the middle? Yeah, because you want a copy of every single one, right? Now, I made some red and some blue and some green. Why did I do that? What was I inferring by that? Yes? Homologous pairs, yeah. So when we learn meiosis, remember, we would talk about the big green and the big blue would line up together, and the little green and the little blue would line up. Those would be homologous pairs. You only do pairings when you're getting ready for sex, right? That's the only time when you get the pairs, but that makes sense, right? Pairs for sex. So when you're getting ready for sex, you have to get down to half as many. That's when they don't line up single file. That's when they line up with a partner. Because in meiosis, the first division is separating homologous pairs, and then the second division is separating the sister chromatids, right? So pairing only before sex. If you're doing asexual reproduction, if you're making an exact clone, everybody lines up and everybody lines up in the middle of that cell. And then all of you're going to have microtubules that are going to start to extend across, right? And these microtubules are going to pull these sister chromatids apart from each other. And when that occurs, right, then you're going to start to form a nuclear envelope around them, and a nucleolus forms, and you'll have identical cells, again, each cell with 4 chromosomes in it. Do you remember about 2n and n? So if this 2n cell, right, equals a total of how many chromosomes? 4. Then what would be the n number? 2, right? 2 times 2 is 4. When would I have just an n number? If I had a sperm or an egg. Then I would put an n with an n, diploid, zygote with 4 chromosomes again. Okay? Check with your bio buddy. See if they got what I just was pointing out to you right there. Okay, next. Can you name and describe 3 different ways meiosis contributes to genetic variety? See if you can come up with the 3 on your own first. Think about it real quick. All right. So what are the 3 different ways meiosis contributes to genetic variety? Independent assortment, I like that. What else? Independent assortment, crossing over, and what else? Fertilization. Okay, now the question is, do you know what those 3 things are? Okay, so when you do meiosis, remember we said we would line up big green with big blue and little green with little blue, right? That's synapsis. You're forming a homologous pair of chromosomes. Each homologous pair lines up independent of the other pairs. All it has to happen is the pairs have to be in the middle. So it doesn't matter if all the greens are on one side and all the blues are on another side. It doesn't matter. That's independent assortment. So that means you can get any combination. You could get, in your gamete, you could get a mixture of green and blue traits. Does that make sense? Since you're going to mix those up anyway when you want to. That's independent assortment. Crossing over also happens during synapsis. When those homologous pairs are together, the sister chromatids could overlap. It's okay because it's the same type of genetic variation. It may be coding for different colors of eyes or different colors of hair, but it's still coding for hair and it's still coding for eyes, so that's all good. It just mixes it up. Do you remember how we talked about hair color and eye color traveling together? So most people with dark hair have what? Dark eyes. And people with light hair have light eyes. Okay? When you have somebody with brown hair and blue eyes, then that is evidence of somewhere in their ancestral past they did what? Crossing over. My mom, I'm supposed to have brown hair, or auburn hair, and I have blue eyes. My mom had brown hair and blue eyes, and my Grammy had brown hair and blue eyes. So that means way back there that event took place. Does that make sense? Okay. Fertilization. Why do we say that? How does that contribute to variety? Yeah, you don't know what sperm is going to fertilize what egg. Now, why is variety so important? Yes?
It helps for evolution.
Yeah, for evolution, yeah. It makes it so that there's variations amongst the offspring. Some may be better fit than others. Remember, fitness is the degree to which you are adapted to your particular environment. So the variety, you need to have variety if you're going to look at something like natural selection. What word do you hear in natural selection? Nature selected. Good. Okay. Next, I said, can you describe the similarities and differences between mitosis and meiosis? Meiosis 1, meiosis 2, cytokinesis in plants and animals. Okay. Mitosis and meiosis. Mitosis, how many divisions do you go through? 1. Okay, you take that one cell, copy all of its chromosomes, separate them from each other. So you have 2 cells now with 2 sets of information. But in meiosis, you're getting ready for sex. And so you're going to have 2 divisions. The first division is going to separate the homologous pairs. So now you have two cells, okay, with no homologous pairs in them. Then the second division, you separate the sister chromatids. So meiosis 1 and meiosis 2, we just said, you have separation of homologous pairs versus separation of sister chromatids. Which one does independent assortment occur in? What is it? Meiosis 1. Does independent assortment occur during mitosis? Why not? Yeah, there are homologous pairs in there, but they're not lined up with each other, right? Everybody's in single file, right? So that's why independent assortment is going to occur during meiosis 1. At what stage of meiosis 1? Metaphase 1. What does happen during prophase 1 of meiosis? Crossing over. Okay, good. Cytokinesis in plants and animals. In animals, you're going to form a what? Cleavage furrow. But in plants, you've got to build a cell wall, cell plate, and then it becomes a cell wall, right? Because they don't have that flexibility like animal cells do. So they've got to build a cell plate and cell wall. Okay, check with your bio buddy. Make sure you're feeling strong and see if you have any questions on that. Do you need me to go over? Okay, questions you want to ask me? All right. Can you describe the similarities and differences between oogenesis and spermatogenesis? Well, I wonder what you're making in spermatogenesis. Sperm. Okay? Oogenesis, what are you making? Eggs. Do you remember the key difference between spermatogenesis and oogenesis? Polar bodies, right? Spermatogenesis, that you're most adaptive if you make a plethora of sperm. And that's exactly what you do. You don't have to pack anything. All you got to get is that nucleus to that egg. So they're small, they're fast, they don't grow. Okay? In oogenesis, you have time between ovulation and implantation into the uterus. And so there has to be nutrients there to sustain it until they get from the ovary to get implanted in the uterus. So what you do is when you do cytokinesis, you do it unevenly. So you will make the nuclear material is even-steven, right? Because you're going to do meiosis, you're going to separate the chromosomes out, but the cytoplasm is uneven. And as a result, you get one large egg and a polar body, and that little tiny polar body may or may not undergo meiosis 2. And in the one large egg, when it undergoes meiosis 2, again, one large egg and a little tiny polar body. So when females do meiosis or oogenesis specifically for females, we just make one egg and 2 to 3 polar bodies. But when men do spermatogenesis, they make 4 sperm. Okay, and that is to allow you to have nutrients to get to the implantation in the uterus. That comes into play when we talk about development, because primarily the things in the cytoplasm of a developing zygote are going to be from who? The mom, the egg, right? That's whose cytoplasm it's going to influence the development. All right. Any other questions on chapter 5 that you want me to address? Okay, chapter 22. Can you name the structures of fertilization? So let's do that song. Here comes Mr. Happy Sperm. And what does he have on his head? Acrosome. What's inside the acrosome? Enzymes, right? What are the enzymes for? To digest the zona pellucida, which is the covering around the egg, yeah? The sperm fertilizes the egg and you form a diploid zygote. That zygote undergoes cleavage, which is a lot of mitosis with very little growth during G1. And you form a solid ball of cells called a what? Morula, okay? Solid ball of cells. Then you pump in what? Salt. Who follows? Water, and it forms a blastula. And a blastula is a hollow ball of cells. Good. And what day does this happen? Day 5. So now you have a blastula. Okay, then you're going to fold, invagination. Okay, and you're letting out, it's like I told you, like those red rubber playground balls, if you let the air out of them, you punch it out, like you could wear it as a hat. Okay, then you're going to form a gastrula, which is a layered ball of cells. Do you remember what the outermost layer is called? Ectoderm. And the innermost layer? Endoderm. And then later the middle layer forms, which is the mesoderm. We know what's going to become from each of those layers. They already have studied that. What comes from the ectoderm? Your skin and your, not your hair. nervous system, and the lens of your eye, right? Things on the outside, skin, nervous system, lens of the eye. From your endoderm, what comes from that? If you go inside your mouth, right? What two systems are you going to run into when you go inside your mouth? Digestive and respiratory. Your digestive and respiratory systems, the linings of those, and any associated organs. Everything else comes from the middle layer, which is called your mesoderm. And if you just hold open your arm, or hold out your arm, what do you see in the middle of your arm? Your muscles and bones. And whatever other systems that are not nervous, respiratory, and digestive, right? All the other ones come from the mesoderm. Okay, so gastrula, and then the next, so we did morula, right? Blastula, gastrula, and then nerula. Nerula with an N, that stands for that's when your nervous system actually develops from your ectoderm. Tell your bio buddy, are there some things you need to review in that? Oh, yes, I totally forgot. Thank you. Sorry. Okay, so I dropped off. Let's go again. Here comes Mr. Happy Sperm. What's on his head? Acrosome. What's in it? Enzyme. Enzymes are for digesting the zona pellucida, like a jelly coat on the egg. It forms a diploid zygote. Cleavage, 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 and you form a solid ball of cells called a morula. Pump in salt. And who follows? I don't know what I was doing. Water, and you form a blastula. A blastula is a hollow ball of cells. Then we do invagination, and we form a gastrula, which is a layered ball of cells. Now, the cavity inside the blastula was called the what? Blastocele. Now, when you form the gastrula, the opening to the gastrula is called the blastopore. The cavity inside is called the archenteron, right? That's the cavity. And I think like my thumbs make an archenteron. The opening is the blastopore. For us, the blastopore becomes our anus. That's why I have you go blastopore. But for everybody, not everybody, I don't know what that's saying. But that's because we're deuterostomes, but protostomes, like mollusks and insects, then the blastopore becomes their mouth. Okay? You have a gastrula, and then a nerula is when you form your nervous system. Now, do you remember a notochord? The notochord from the mesoderm is what gets the spinal cord to do this invagination and form itself, and that's called induction. When one tissue influences like peer pressure on neighboring cells, it's called induction when it does that. Okay? All right. Can you name and describe all the stages of the embryo that we just did? Okay. Can you name and describe the layers of the gastrula? Yes, we just did that too. Can you differentiate and give an example of induction and morphogenesis? We can. Okay, so induction I talked to you about when the notochord influences the spinal cord to form right above it. That is an example of induction. Then I talked about the lens of the eye. Optic vesicle inducing the lens to form, and then the lens getting the optic vesicle to become the optic cup. Does that sound familiar to you? Am I speaking AP right now? I'm speaking AP. Okay, just ignore that. Okay. Morphogenesis. You're like, stop talking about foreign language. Morphogenesis is the movement of tissues in order to become, like your eyes come down and move from the side of your head to the front of your head. That's morphogenesis. And it's the folding and development of your body parts. Can you describe how mitosis and meiosis contribute to the stages of fertilization development? We can, because we know meiosis creates those sperm and egg, right? And once you form a diploid zygote, you're going to be doing what? Mitosis. Until in your gonads, you're going to be doing meiosis, which then makes sperm and egg, and sperm to egg get together by fertilization, and then you do mitosis, right? Circle of life, Simba. Okay? That's it. Chapter 20. Any other questions you want to ask me about that one? Okay, chapter 23. Can you name Mendel's 3 laws and describe them as well as provide an example of each? I bet we can. The law of dominance. That would be like a tall, the trait for tall is dominant over the trait for short. Okay, so law of dominance, law of Segregation. Law of segregation says if you have a big T and a little t, which could be on a homologous pair of chromosomes, what we just learned about in meiosis, that big T becomes separated from little t. You only get one copy of the T-ness in the gametes, right? And then what's the third law? The law of independent assortment. That means you could be yellow and round, or you could be yellow and wrinkled, right? You could be tall with green leaves or tall with yellow leaves, right? Those are different traits, and if they're on different chromosomes, they can assort independently. Okay, those are Mendel's 3 laws. And there are exceptions to all of them. Somatic cell and a gamete. Gametes are sperm and egg. Somatic cells are body cells. Phenotype and genotype. Okay, the genotype codes for a phenotype. So big T is a genotype, and that would code for tall. But big T, little t, a different genotype, would also code for tall. Okay, little t would code for short. Okay, the phenotype is the expression of the genotype. Right, it's how it appears. Homozygous and heterozygous. Homozygotes mean both of your alleles are the same. Big T, little t, little t. Big I, A, or little I. Those are homozygous. In the zygote, it's the same. Heterozygote is any kind of hybrid. Big T, big R, big IA, big IB, right? Those would be heterozygotes. Blood types, okay? I just kind of been going over that a little bit. So you could have big IA, that would be type A, but so would that, right? How do you have type O blood? And same thing for B. How do you have type O blood? Little I. When I say little I, that's a genotype. When I say that that is type O, that would be the phenotype. And AB blood would be big IA, what? Big IB. And this is an example also of multiple alleles, okay? Because you have big IA, big IB, and little I. It's not just tall and short, right? We have 3 options here of alleles, which give us a lot of options on phenotypes. Incomplete dominance. Do you remember what that one is? Red flower crossed with white flower makes pink flower, okay? You know the best way to get this genetics, to make sure you know how to do that, is to do the practice problems at the back of the book, or and throughout the chapter, right? And all the answers are there. Sex-linked traits. Sex-linked traits, remember, you have to look on the female, right? You could have X and X little n, okay? You could have different traits that are on the X chromosome, but you don't see anything on the Y chromosome. And that's why, for instance, like color blindness, males only have 1 shot at their X because they are XY. If it happens to be a bad X, they're colorblind. Whereas girls, we have 2 shots at our X. So both of them, if it's a recessive trait, both of them have to be bad in order for us to be colorblind. So when you do those kinds of crosses, you need to put the trait on the X. None that you have to put on the Y. There are traits like that, Hollandric traits, but I won't give you any questions like that. Polygenic inheritance, like your intelligence, you have A's, B's, Q's, R's, S's. Poly means many. Genic is genes. Many genes working together for that one trait. It's not just smart and stupid. You know, it's not like that. Look at the skin color in this room. There's all different kinds of skin colors because that's a polygenic trait, right? All different heights. It's not like we're tall or short. You know, we have all degrees and ranges. Those are really good examples of polygenic. Codominance, AB blood would be a good example of codominance. If I had a black bunny and a white bunny, and they had baby bunnies, what would some of their hair is black and some of their hair is white be examples of? Codominance. But if I had a black bunny having sex with a white bunny and every single hair was gray, that would be what? Incomplete dominance. Okay. Red flower crossed with a white flower makes pink flower. Straight hair crossed with curly hair gives me wavy hair. What was that one? Incomplete. Okay, because it looks like blending. It's not blending, but it just neither one of them is dominant over the other. Questions you want to ask me about this page? Okay. Can you identify and give specific? Oh, we just did. We just did. We were already on top of it. Can you do a Punnett squares with one trait and give genotypic and phenotypic results with 2 traits or 3 traits? Do you remember how to do that? Your Punnett squares? Anybody need me to go over that? Can you explain why a test cross is necessary? Do you remember test crosses? Yes.
A test cross is necessary because otherwise there's no way to determine the genotype of the parent.
Yeah, I didn't hear the very first part, but it's when they're displaying a dominant phenotype, and you don't know if they're homozygous dominant or heterozygous dominant. So what do you cross them with? Homozygous recessive, right? If I had a person who had brown eyes, right, I don't know if they're brown-brown or brown-blue, right? I don't know if they're carrying that blue-eyed gene. So who would I have them have sex with? Somebody who had blue eyes. Because every single time the blue-eyed person, in order to be blue-eyed, they have to be homozygous recessive for blue eyes. So they're always, all their gametes are going to have blue in them. So if they have any children with blue eyes, then you know the unknown genotype has to be a heterozygote, right? Does that make sense to you? Okay. But if they all have brown-eyed babies, then probably they had brown eyes. Brown, sorry. Do the practice problems in the chapter, and at the end of the chapters and answers in the back of the book. I already told you to do that. And you don't have to do your additional objectives. You won't have to know those on there. Okay? I think that's it, you guys, for Unit 5. Identify those areas where you're weak. Get studying on those, right? And tomorrow we're going to do Unit 6. And then Wednesday we're doing Unit 7. Thursday I go see Summer. And then I'll be available at lunch if you want to talk to me. And then Friday we do anything in Unit 8 that you want to review. Okay? Questions or concerns? Do you know that I love you? Do you know that I want you to get 100% on your final and a good grade? Okay, make good choices. And have a piece of toast.